some unexpected findings are possible, as for instance with so-called masked mycotoxins, where the bondage of deoxynivalenol to glucoside, which I've already presented in my previous slide, was for the first time detectable in actually Franz Bertiller's PhD thesis, and he actually found out that up to 45% of deoxynivalenol in wheat samples occurs as additional conjugated ketone-3 glucoside, which can certainly have implications also for food safety, especially in case the parent mycotoxins might be released after enzymatic hydrolysis in the gastrointestinal tract. And this can also be used for targeted analysis of fungal metabolites. So what we did is actually adding zearalenone to Arabidopsis thaliana and see what is the capability of a plant to metabolize a mycotoxin. And actually, you see, even with time mass spectrometry and precursor ion scan, 97% of the zearalenone was metabolized within 24 hours with a great variety of different metabolization products, D-glucosides, malonyl glucosides, sulfates, zearalenols, and so forth. So quite an amazing capability of plants to degrade, to metabolize mycotoxins to other components than the usual masked mycotoxins which we have got in mind, and which actually is a research field on its own. And that actually leads us towards metabolomics, where we combine our sample preparation with high-resolution mass spectrometry and gas chromatography, so LC, high-resolution mass spectrometry, combined with gas chromatography with MS, to cover the whole set of all possible small molecules, secondary metabolites which are formed by an organism. That is actually done by Rainer Schumacher. He's the head of our metabolomics group. You get 2 million peaks of masses. You need bioinformaticians in order to reduce that to a reasonable size of possible compounds. In this case, actually 9 bioinformation compounds were found with actually a new generation, so to speak, of mycotoxins which we have detected. The so-called, we metabolize deoxynivalenol on wheat, and after metabolization of deoxynivalenol in wheat, we found deoxynivalenol glucosides. We found some 5% of remaining deoxynivalenol, but we found actually DON SGSH, which is actually glutamyl, cysteine, and glycine, the so-called DON glutathione pathway derived metabolites, which is actually a new discovered range of masked mycotoxins. We don't know anything about their toxicity. I think they are pretty unstable, but nonetheless, that shows another detoxification potential of plants to metabolize deoxynivalenol to less toxic compounds, because xenobiotics like pesticides, which is used to treat grass, your lawn in your garden, does also include so-called pesticides or herbicide saverness, which kind of activate the glutathione pathway in order to make the pesticides, the herbicides, less toxic for the grass. So that's actually a well-known phenomena which has so far not been detected in the mycotoxin community. And what I wanted to point out here is the area of biomarkers of exposure. I have already mentioned that now with the sensitive methodology which is available, we are now able to also detect deoxynivalenol and its glucuronide, so the oxidized form of glucose in urine. So that is actually quite an interesting experiment. So when you exposed to a regular diet, you quite likely, if you're an Austrian, find levels of 30 to 100 microgram per liter deoxynivalenol glucuronide in your urine, and Benedict Wartz, who performed his PhD thesis in this area, then actually restricted himself to a couple of days living in austerity, only consuming potatoes, which were free of deoxynivalenol, and you see actually the black baseline that actually, if he is not consuming any potentially deoxynivalenol containing diet, there is also no glucuronide, no biomarker in his urine. So that can actually be used to study these biomarkers of exposure in developing countries where there is a more likely exposure to multiple mycotoxins. And actually, from here is just an example from a patient in Cameroon. 6 mycotoxins and metabolites like nivalenol, DON glucuronide, DON aflatoxin 1, FP1 were detected simultaneously. That's not the latest study. There's actually a more recent one, but this actually is a very impressive picture where you see actually in the urine where we are able to determine the mycotoxin itself and its glucuronide kind of transformation, detoxification products produced in the liver. By the way, as some of you might know, cats do not have this possibility of detoxifying in the liver to their glucuronide. That is why they are much more susceptible to mycotoxins.